Good afternoon, everyone, and welcome to this first part of this COVID-19 webinar. As already mentioned by my colleague Sylvia, my name is Julianne Bobela, and thanks to her, you already know quite a few things about me, so I can skip this formal introduction. I would, however, like to thank you for joining in this online presentation today, and though I won't be able to see you, it's a pleasure to spend the next half an hour with you, where I will talk about the epidemiological background of this nowadays very famous and the least we can say quite troublesome new virus, as well as introduce you to the type of tests currently available for the diagnosis of COVID-19 patients. Please, at any time during the presentation, feel free to send us your questions using the chat that you can find on the webinar's webpage. And at the end of the webinar, we will try to address all of them and provide you with an answer. So let's get started with a quick chronological overview of what happened these last few months. But before this, let me shrink myself so you can see the actual presentation. There we go. So everything started in December 2019, where unexplained viral pneumonia cases were identified in Wuhan in China. And by January 12th, a new coronavirus was identified as causative pathogen for this disease, and its sequence was then added to the GenBank sequence repository. January 20th of this year, it was then confirmed that the virus can spread between people, and 10 days later, the WHO declared the coronavirus outbreak as a public health emergency of international concern, making it the sixth public health emergency after H1N1, polio, Ebola in West Africa, Zika, and Ebola in Democratic Republic of Congo. By that time, in China, there were more than 7,500 confirmed and more than 12,000 suspected cases of the disease, and it had already spread to 18 other countries. By February the 12th, this virus was then renamed Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2, and the related pneumonia disease as Coronavirus Disease 2019, or COVID-19. March 11th, the WHO then declared the coronavirus outbreak to be a pandemic, and I guess since then, most of you have spent quite some time at home practicing social distancing and trying to stay away from coughing people. During these days, last 2.5 months, things have also evolved quite a bit. And as of today, the disease has spread to more than 200 countries and infected close to 6 million people and resulted in almost 360,000 deaths globally. Also in the scientific community, plenty of things have been going on, and there are a lot of different publications that come out almost on a daily basis covering this COVID-19 topic, as you can see here with the hits that are retrieved using these different keywords. Now, the question is, what is this coronavirus exactly? So the family of coronavirus has first been described in 1966 by Tyrrell and Bino, and it contains the largest enveloped single-stranded positive sense RNA viruses that have a diameter that ranges from 60 to 140 nanometers. These viruses, they all have spike-like projections on their surface, which give them a crown-like appearance, hence their name coronavirus. The Coronavirinae subfamily contains the alpha, beta, gamma, and delta genus, with the gamma and delta genus predominantly infecting birds, but they also can lead to infection of mammals with, for example, whales and pigs. And so far, 6 coronaviruses had been identified in the human and the alpha and the beta coronavirus genus, with the SARS-CoV and the MERS-CoV, and now the SARS-CoV-2 being the most severe types that can lead to lower respiratory tract infections and acute respiratory distress syndromes, which can cause patient death. So this novel SARS-CoV-2 virus is a positive sense RNA virus, which belongs to the beta coronavirus genus. The question now is, where did this new coronavirus exactly suddenly emerge from? And bats are known to be the natural reservoir, and they might be the original host of this virus. However, they are unlikely to be the animal that is directly responsible for transmission of the virus to the human, although this direct route of transmission is not excluded. But most likely the transmission from bat to human happened via an intermediate host, and this intermediate host plays a critical role in the cross-species transmission, as it can facilitate increased contact between the virus and the new host and enable further adaptation of the virus, which is necessary for an effective replication in the new host. Excuse me, there we go. So the SARS-CoV-2 virus has a single-stranded positive sense RNA genome that is approximately 30,000 nucleotides in length, and the genome encodes for 27 proteins, including an RNA-dependent RNA polymerase, and 4 structural proteins, which are the spike glycoprotein S, the M protein, and the N protein and E protein. In general, the spike protein of coronavirus is divided into the S1 and S2 domain, in which the S1 is responsible for the receptor binding and the S2 domain is responsible for cell membrane fusion. The other 3 structural proteins are more conserved than the spike protein, and they are necessary for general coronavirus function, which involves encasing the RNA in protein assembly, budding, envelope formation, and pathogenesis. Now, viruses, they don't just wander into the cells they infect. They actually first need to attach to a specific receptor found on certain type of cells to be able to infect cells. Excuse me. There. So for the SARS-CoV-2, infection of the cell happens through binding to the angiotensin converting enzyme 2, or AC2. So the virus binds to this receptor, letting itself into the cell, where it then replicates itself and can infect other cells around. Usually AC2 is found on lung, kidney, heart, and gut cells, and recently researchers also discovered the presence of AC2 receptors on the cells in the nose. So as you can see, the receptor responsible for the SARS-CoV-2 infection of cells is quite widely distributed among cells of the human body, which might explain the broad spectrum of symptoms that are observed for COVID-19 disease. Mean incubation period between the infection and the appearance of the first symptoms is around 5 days, but it can also be as long as 19 to 24 days. Between this quite long incubation period and the high transmissibility of the virus, this can explain why this has become to such a difficult case to handle. So the transmissibility of a virus is reflected by its reproduction number R, which represents the average number of new infections that are generated by each infected person. So the initial constant of which is called the basic reproduction number R0. And so the larger the R0 is, the stronger the transmission potential of the virus. An R value that is above 1 indicates that the outbreak will be self-sustaining unless effective control measures are implemented, while our R value, which is below or lower, smaller than 1, indicates that the number of new cases will decrease over time and the outbreak will eventually stop. So for the SARS-CoV-2 virus, the R0 was estimated by several groups to be 2.2, 3.11, or 2.68, depending on the group that has analyzed this value. And however, one can say that based on this R0 value, that the SARS-CoV-2 has a very high transmissibility potential, and that it consequently has the ability to cause outbreaks, as we have seen. Now, the disease is mainly spread by respiratory droplets and contact between persons, and at present SARS-CoV-2 infected patients are the main source of infection, producing a large quantity of virus in their upper respiratory tract. This virus also leads to mild symptom in certain patients, and also the incubation time is quite long, which means that some carrier of the virus, they keep on having routine activities leading to the spread of the infection, and also asymptomatic carriers can be found, which are source of infection. So all of this between asymptomatic carriers, quite a long incubation period, and a high transmissibility led to the situation where we are currently in, which means quite a drastic outbreak of this virus. So let's talk now a bit about the symptoms that can be caused by this SARS-CoV-2 virus. So common symptoms observed in COVID-19 patients are headaches, loss of sense of smell and or taste, a dry cough, sore throat, mild breathing difficulties, gastrointestinal issues, including diarrhea, and in lots of cases, fever, and also general body aches and fatigue. In a few cases, these symptoms then become more severe, and one can observe heart failure, pneumonia, respiratory failure, kidney failure, high fever, or sepsis, septic shock, and even death of the patient. So as this virus has a very high transmissibility and also leads to quite a severe disease with the possibility of fatal outcome, it is very important to control its spread amongst the community. And diagnostics can play an important role in the containment of the COVID-19 outbreak, enabling the rapid implementation of control measures that limit the spread through case identification, isolation, and contact tracing. However, as the symptoms expressed by COVID-19 patients are non-specific and quite broad, they cannot really be used for an accurate diagnosis. So besides syndromic testing, a CT scan of the patient's lung is often used to confirm COVID-19 diagnosis. As you can see here on this CT scan, in the COVID-19 patients, there is the accumulation of fluid in the lung often leads to these signature, which are called ground glass. And so the combination of the symptoms and the appearance of these traces on the CT scan, they can actually sometimes help to confirm the diagnosis of COVID-19. However, molecular techniques are more suitable than syndromic testing and CT scans for accurate diagnosis because they can specifically target and identify the COVID-19 pathogen. The development of molecular technique is dependent upon understanding, first, the proteomic and genomic composition of the virus and the induction of changes the virus can trigger in the host during and after infection. So molecular techniques used for the diagnosis of the COVID-19 disease, they include testing for the SARS-CoV-2 virus itself or testing for response of the host, which after being infected then starts to secrete some specific antibodies to the virus itself. Some tests are point-of-care tests, meaning that results may be available at the testing site in less than 1 hour, and other tests must be sent to laboratory to analyze and process. That takes 1 or 2 days once received by the lab. So one molecular technique for COVID-19 diagnosis is the nucleic acid testing through PCR, which is today the gold standard method for the clinical diagnosis of COVID-19. So it's the nucleic acid testing in combination with CT scans are the most widely used for the diagnosis and screening of COVID-19 patients nowadays. During this testing, we are actually looking at the SARS-CoV-2 viral RNA through PCR, and a number of reverse transcription polymerase chain reaction kits have been designed to detect specifically the SARS-CoV-2 genetically. So what exactly is being done during this test? So it is first through nasal swab. You can take some extracts of some samples that where you then do a RNA extraction to obtain the RNA of this sample. Then through reverse transcription, you proceed to the transformation of this single-stranded RNA into cDNA of the virus. Then using very specific specifically designed primers for the SARS-CoV-2 virus, you manage to amplify regions of this cDNA by PCR or by real-time PCR, which allows you to obtain quantitative results. And then using dye molecules, you can detect this amplified viral cDNA and determine if a patient has been infected by the virus, meaning that you will have you will obtain here a signal, or if non-infected, you will have no DNA amplified. So in a one-step assay, reverse transcription and PCR amplification, they are consolidated into one reaction, and this assay format can provide rapid and reproducible results for high throughput analysis. But the challenge is the difficulty to optimize the reverse transcription and amplification steps as they occur simultaneously, which leads to lower target amplification generation. In the two-step assay, the reaction is done sequentially. So first there is the reverse transcription and then the PCR reaction. And they are done in separate tubes. This assay format is more sensitive than the one-step assay, but it is more time-consuming and requires optimizing additional parameters. Now, given the variability in the viral loads and negative test results from respiratory sample does not rule out the disease, and these negative results could result from improper sampling techniques, low viral load in the area sampled, or mutations even in the viral genome. So the advantages and disadvantages of using RT-PCR for the diagnosis of COVID-19 patients. So in the pro section, this is an extremely reliable tool when carried out properly. And as disadvantage there is the availability of PCR reagent kits, which has not kept up with the demand recently. And also some smaller hospitals, they actually lack the PCR infrastructure to accommodate high sample throughput. RT-PCR relies also on the presence of detectable SARS-CoV-2 in the sample collected, which after a longer period of time of infection, we know that the presence of the virus actually decreases. Also, this technique needs trained users which are required to perform the tests. And a disadvantage also is that this process that is usually sent to a laboratory can take up to 1 to 2 days. So these last 2 points, they have actually triggered the development of new tools for diagnosis of COVID-19, which would allow COVID-19 patients to be tested more rapidly, and also by hospital staff that is not necessarily trained in RT-PCR. One of these methods is the nucleic acid test using isothermal amplification, where a nasal swab can be put directly into a tube and in combination with specific reagents, and here a combination of 4 to 6 SARS-CoV-2 specific primers. A result of this test can actually be obtained in less than 1 hour, and with an incubation temperature that is around 60 and 65 degrees, so which does not require a thermocycle. And then the test, the readout can be then usually visualized through turbidity, the color, or the fluorescence. Now, the advantages of this type of test are that it is simple to operate, it's easy to visualize for detection, and it does not require a thermocycler. So basically, people in a hospital can perform this test quite rapidly, and don't need to be trained for too much for such activities. The disadvantages, however, are the challenges of optimizing the primers and the reaction conditions, because of course here we need really specific primers, because the reaction conditions are a bit less favorable in this aspect. As previously mentioned, another way of directly testing for the presence of the virus itself is by detecting not the viral RNA, but specific proteins of the virus. So these are called antibody-based immunoassay techniques, where in laboratories there are antibodies that are being developed to specifically recognize antigens of the SARS-CoV-2 virus. And these antibodies, they can then be directly linked to a reporter conjugate, or one can use a secondary antibody with a reporter conjugate, which would allow to amplify as well the signaling. This testing here is potentially a very rapid detection method, but the changes in the viral load over the course of the infection may make the viral proteins difficult to detect because unlike for the PCR, there is no amplification step of the signal itself. So basically, in the PCR, you first start with RNA, which then you amplify, and then you visualize the amplified RNA. And here you have a certain amount of proteins that are expressed, and you don't amplify first these proteins before being detected by your antibodies. Antibodies also need to be really specific to these proteins, not to detect other type of coronaviruses. And if you then want to amplify a bit the signal, you could use the secondary antibodies. But here the drawback is as well that these must be really specific and not bind to other type of proteins on the surface of the virus, not to give too much of a background noise. Now, all of these test methods, they rely on the presence of detectable SARS-CoV-2 in the samples collected. And however, we know that over time, the viral load decreases to undetectable levels. And after a while, it is therefore not possible anymore to tell if a person has been in contact with the virus or not through these detection means. For this reason, it is important to develop tests that allow to detect changes in the virus, changes that are triggered by the virus in the host during and after infection, which basically means looking for antibodies that are produced by the immune system of the host. So antibody blood tests, also called antibody tests or serological tests, check your blood by looking for antibodies which show if you had previously been infected with the virus and the body has responded to it by producing an immune response, which means that T cell, they start to secrete antibodies directed against the virus it has been exposed to. These antibodies are proteins that help fight off infections, and it takes time for such antibodies to actually be created. Now, depending on when someone was infected and the timing of the test may not find antibodies in someone with a current COVID-19 infection. However, in contrast to the RNA tests, they can be extremely useful in determining whether someone has previously been infected with the new coronavirus, but no longer has the virus present. And since people that have suffered from COVID-19 disease, they might express symptoms of this disease over quite a long period of time. Sometimes it could be really helpful to know if a patient that comes with some symptoms, if they had been previously in contact with this disease without developing severe symptoms. Now, antibody tests should, however, not be used to diagnose someone as being currently sick with the COVID-19. One complicating factor may also be that the different people, they can have different antibody responses to COVID-19. For example, an individual with severe disease seem to develop higher antibody levels than individuals with mild or asymptomatic disease. And as a result, a test for antibodies developed using blood samples from individuals with severe COVID-19 may not work as well in detecting antibodies in people with a mild or asymptomatic version of the disease, where there are fewer antibodies to detect. Now, concerning this diagnostic tool, the advantages are here that there is a larger window of time for indirectly detecting infection with SARS-CoV-2. This tool is also very useful in determining whether someone has previously been infected. The disadvantages are the potential cross-reactivity of the SARS-CoV-2 antibodies with antibodies generated against other coronaviruses. Hence, the importance actually to really develop antibodies which are highly specific to this type of coronavirus. And this tool is also not really much of a help in confirming if someone has COVID-19 in the first few days of infection, since the body has not had time yet to create an immune response and antibodies against this virus. Then also one disadvantage is that different people can have different type of antibody response to COVID-19, meaning that one developed test might actually not detect all types of, might not be useful to detect all types of antibodies that were directed against the COVID-19 virus. So as you can see here, there are plenty of tests that are currently being developed for the diagnosis of COVID-19 people, which rely on different biomarkers, type of technology, type of clinical samples that are required. And I guess plenty of labs are currently trying to develop their own tools with regard to this situation right now. So according to the WHO, the immediate priority for COVID-19 diagnostics research is the development of nucleic acid and protein tests and detection at the point of care, and which means that these point-of-care tests are used to diagnose patients without sending the sample to centralized facilities, thereby enabling communities without laboratory infrastructure to detect infected patients. So lateral flow antigen detection for SARS-CoV-2 is one point-of-care approach, for example, under development for diagnosis of COVID-19 patients. So, and as you might all be aware, there is, however, usually a big gap between the number of diagnostic technologies that are being developed and the ones that actually reach the market, with the phase 1 and 2 typically occurring in an academic setting, while phase 3 and 4 occur in company after commercial transfer. Now, most diagnostic technology are at the proof of concept stage, and few are in phase 3 that can be quickly adapted for the diagnosis pathogens in new outbreaks. The advancements of more phase 2 technologies into phase 3 would increase the number of approaches for detecting new pathogens. Now, it is really important, however, to, during all of these phases of the development of the diagnostic tool, to really consider all the regulatory implications of the development of such tools. And we will, of course, in the next part of this seminar talk about these aspects of the development of new diagnosis tools. So these just are the references that I've used for this presentation, where I also took the freedom to choose some pictures as well to show you today during the presentation. And I would like to thank you very much for your attention, and I hope that you enjoyed this presentation and that you will enjoy the rest of this webinar, and that you will have plenty of questions by the end of our talks. Thank you very much and have a nice continuation. Bye-bye.